not quite there. Just give me a few seconds.
Okay, so it's also my pleasure to be able to introduce the 2015 Okubo Award winner, who is not a surprise because he's standing up here. So this is the award from the Society to honor Akira Okubo, who some of us were fortunate to meet, and the language of the award is for outstanding and innovative theoretical work for establishing superb conceptual ideas for solving tough theoretical problems and/or uniting theory and data to advance a biological subject. And I think the and/or is in the case of this year's recipient an and. He has done all of these things, and it goes to Joshua Plotkin at the University of Pennsylvania. Josh received his undergraduate degree in math at Harvard and his PhD at Princeton, working with Simon Levin and Martin Novak. He was a Burroughs Wellcome Fellow, a Sloan Fellow, Packard Fellow, and also a Junior Fellow at Harvard, and is currently Professor of Biology and Computer and Information Science at the University of Pennsylvania. He's worked on a variety of problems, ranging from species area curves, the evolution of cooperation, evolution of language, and I'd say most deeply and most recently, how fitness landscapes and genetic architecture are shaped by evolution and in turn shape evolution itself. For me, Josh's work really parallels his personality, which I like to characterize with apologies to Kipling as insatiable curiosity. He's somebody like the elephant's child, if you remember this story, who's willing to ask questions and take risks. And I think in his case, he always receives these surprising answers that with themselves have surprising rewards along the way. So we have for him a ceremonial plaque. And within this envelope, a ceremonial check, which he can enjoy as he wishes. Great, so he's going to teach us about the role of epistasis in molecular evolution.
Good morning. Is this on? Okay, well, thank you very much for that lavish introduction, Fred. And I want to start right off the bat by thanking the Society for choosing to award me the Okubo Prize. It's something I'm very proud of and I certainly appreciate and I'm honored by. I'm going to talk today about a series of projects over the years that have come, at least in retrospect, to realize all revolve around this question of how epistasis is involved in evolution at the molecular scale. And by way of introduction, to introduce this problem, I'll have a few comments about evolution in the most simple, caricatured way. Darwin told us, for example, that in a new environment, a population will tend to acquire and retain those variants that increase organismal fitness. And that is sort of shown by this sort of cartoon view of a population increasing fitness over time. But as revolutionary as it was, Darwin's theory of evolution was largely qualitative. What Darwin didn't tell us are all the things that I'm interested in. In particular, how quickly will a population tend to adapt, and by what pattern will it adapt? And most importantly, how does evolution and the process of adaptation depend upon the structure of the so-called fitness landscape, which is shown in cartoon form here? I'm particularly interested in how evolution depends upon features of the fitness landscape known as epistasis. Epistasis describes the idea that the effect of a mutation on an organism at one site in your genome may depend upon the status of your genome at other sites. That is to say, there may be some non-independence between different sites in your genome in their effects on fitness. And in the presence of epistasis, we can have fitness landscapes such as this one that have multiple local peaks. Without epistasis, this is impossible. So presumably, independence or non-independence between sites will have a big impact on the process of evolution. But exactly what impact it has is anyone's guess. And so I'll talk today about approaches to this problem. The first part that I want to talk about involves studying the role of epistasis in the evolution of proteins. I'll describe some viral proteins and also describe some in silico proteins. And then, at least in the second half of my talk, I'll finally turn to the evolution of actual organisms, or at least microorganisms, and I'll discuss how we can seek to infer the role of epistasis from experiments, and also how we can fail to infer epistasis from experiments in some interesting ways. So, starting with viruses, I have had for a long time an interest in using viruses as just a wonderfully naturally occurring evolution experiment that occurs right in front of us, especially RNA viruses that evolve rapidly. So here shown is the influenza A virus, a virus that you guys all have a lot of firsthand experience with, unfortunately. A virus that manages to infect about 1/5 of the globe every year, and nonetheless, most of the people who are infected by the virus recover within about a week, and they are thereafter immune to their infecting strain. And what that means, of course, is that there is tremendous pressure on this virus to acquire, whenever possible, mutations that allow it to escape antibodies that it has recently stimulated or recent strains have stimulated in the human population. And that pressure is mostly mediated on this so-called hemagglutinin protein, which sits on the surface of the viral capsid, elicits neutralizing antibodies, and is therefore the subject of continual positive selection to try to find novel variants that can escape immunity and indeed manage to infect or reinfect hosts every 5 years or so. Of course, as a result of this pressure, it's no surprise hemagglutinin is one of the most rapidly evolving proteins known. Here, just to illustrate that, is a molecular phylogeny of hemagglutinin sequences collected from folks infected with influenza between the years 1985 and 1996. This is a phylogeny that was first made by Walter Fitch. Among several remarkable characteristics of this phylogeny, I'll simply mention the most obvious and impressive one, which is just the sheer amount of evolution that has occurred in this decade time frame. Within this time frame, roughly 20% of the sites in this protein underwent substitutions, amino acid substitutions, which is the equivalent, just to give you a feeling for timescales, of about 10 million years worth of evolution in a typical mammalian protein. And so here we have compressed on sort of a reasonable timescale a whole lot of evolution. And better yet, we know the sequence of essentially every single variant of this protein that reached appreciable frequency in the viral population over time. So we have a complete historical record of an enormous amount of evolution right in front of us. So this, of course, provides a wonderful system for trying to understand what are the forces that shape the course of an evolution, in this case, evolution of a protein, and in particular, what is the role of epistasis in shaping evolution? So that's what I want to ask about this evolving flu protein. In particular, I want to ask, and have been working to ask, whether or not substitutions at different sites in this protein are independent of one another, or are they somehow dependent upon one another through epistasis? And if there are dependencies, can we understand where they are and put them to good productive use? So how would we go about trying to find interactions or epistasis between sites in an evolving protein? So the approach I've taken is ridiculously simple and still seems to work. What we do is we look, for example, at a pair of sites, A and B, in the hemagglutinin protein, and we look along its phylogenetic history at all of those edges where a mutation occurred at either site A or at site B. And we're looking for a pair of sites that have positive epistasis in the sense that a mutation at one site makes a mutation at the second site somehow more beneficial than it otherwise would have been. So there's some synergy between the two sites. Now we reason that if there's some sort of synergy between those two sites, then there will be a special pattern of substitution along its evolutionary history. And in particular, we look and count how much time elapses in synonymous substitutions between mutations at site A and subsequent mutations at site B along the tree. And the basic idea here is that if these two sites are epistatic, positively epistatic, then whenever there's a substitution at this leading site A, there should rather rapidly follow a substitution at the lagging site B. And so we develop a statistic that simply counts. This statistic is complicated only by the difficulties of phylogenetic reconstruction, but all this statistic does for a pair of sites now denoted I and J in a protein simply counts the typical amount of time elapsed between a substitution at the leading site and a corresponding substitution at the lagging site. And we can compute this statistic for any pair of sites in the protein. And then, of course, we need to compare this statistic to some sort of null distribution. And this was, of course, the hard part of this research. We have to devise a null distribution that removes any possible interaction between sites, but nonetheless preserves several important marginal distributions of influenza evolution. So in particular, we wanted to preserve, for example, the total amount of evolution at each site along the tree, because some sites just evolve more quickly than others. We also simultaneously wanted to preserve the total amount of evolution across sites along any one branch of the tree so that the tree has the same shape. In any case, developing that kind of null distribution actually maps to a well-known computer science problem that had been solved, and it can be efficiently sampled. So we can efficiently sample a random null distribution that removes any possible correlation between sites, and then, of course, compare the observed statistics in the data to this null distribution to discover, for example, this pair of sites in flu, hemagglutinin, site 344, a leading site, and site 275, a lagging site. It may be a little hard to see on this screen depending on where you're sitting, but more or less, whenever a mutation occurs at this leading site 344, thereafter, very quickly, a corresponding substitution occurs, for the most part, at site 275. And this pattern occurs multiple times independently across the phylogeny. So there's a very strong, actually, statistical signature of this rapid coevolution of these two sites, which suggests that these two sites are somehow interacting and are together more useful to the virus than they are individually. It's, of course, not just this single pair of sites that we identify. We identify actually several hundred pairs of sites in this one protein with a tolerable false discovery rate that have this, this signature of positive epistasis. And this table alone, this information alone, is already practically useful because it tells us, for example, that if we look at the flu sequences that are circulating the globe today, and the WHO is constantly sequencing flu sequences that are sampled around the world, and we observe a substitution at any one of these leading sites here, then we can predict with what turns out to be about 80% accuracy that there will be a corresponding substitution within about a season or so at one of its corresponding trailing sites. And so that gives us genuine predictive power about the future evolution of this organism. And this is probably the only organism on the planet that we can make specific predictions about its future evolution just by knowledge of what interactions tend to go together in this protein. It's not just, so that's a practical benefit because, of course, we need to choose vaccines, and in some sense, we'd like to predict the future of the evolution of the virus. There's also some intellectual benefits of knowing which pairs of sites interact. We can ask the most simple questions. What is it about the protein structure that lends certain sites to be positively epistatic and not others? And so I'm not going to go through all these. The only thing I'll mention is the most obvious question: how close are these interacting sites in the folded 3D structure? I thought, going into this research, that these interacting sites would just be right next to each other and they'd be physically interacting in the protein structure. And it turns out that although they are significantly closer than random pairs of sites, the interacting sites are nonetheless, on average, about 40 angstroms apart in the 3D structure. So these sites aren't really even close to each other at all. And whatever interactions are mediating this positive epistasis, there are some sort of long-range steric interactions that wouldn't have been apparent from just looking at the protein structure. Okay, I want to tell you one other practical sort of upshot of understanding the role of epistasis in viral evolution that has to do with drug resistance. You guys may know that aside from the flu vaccine, which needs to be updated every few years to deal with flu evolution, we do have in our arsenal one actual antiviral drug for flu called Tamiflu. It's a $1 billion a year drug. It was approved by the FDA, even though it was known, even at the time of its approval, that there's a single site in the neuraminidase protein of the virus, the target of the drug, that confers complete resistance to the drug. It was approved by the FDA because the same resistance site is also lethal to the virus. And so the FDA reasoned, well, this resistance site isn't going to cause any problem. It kills the virus after all. Nonetheless, as some of you may know, by now about half of seasonal flu strains are completely resistant to Tamiflu, also Tamivir. So how in the world did that happen, that there's a resistance, that the one resistance site is lethal to the virus, but somehow there's resistance is widespread? The answer to that problem, Jesse Bloom and David Baltimore reasoned a few years ago, is that there must be epistasis. There must be other sites involved. And so they performed a large experimental screen and identified two other sites, either 222 or 234 in neuraminidase, where mutations at those sites in combination with this resistance mutation produce a viable drug-resistant virus. And so this is, in some sense, through this epistatic interaction, the reason why the virus was able to acquire drug resistance. So why am I telling you this story? This is really Jesse Bloom and David Baltimore's story. The reason is that, and this is also a wet biology story, not a dry biology story. Well, in any case, around the same time that they were performing this large experimental screen, we were developing methods to find epistasis in flu proteins, and when we applied the exact same methods to the neuraminidase protein, the target of this drug, we found, in fact, that amongst the most significant, in fact, the very most significant pairs of sites that are epistatic in neuraminidase are precisely these pairs of sites, 222, 275, 234, 275, that Bloom and Baltimore had identified through an experimental screen, which we can identify just by the pattern of sequence evolution alone without even knowledge of the fact that 275 is a drug resistance site. And so we can understand, in some sense, from the historical evolution of the virus, what interactions are likely to cause drug resistance. And in fact, if you look at this table, we predict a few other leading sites, which in combination with 275, we would suggest would also produce viable drug-resistant viruses. The Baltimore lab subsequently went and tested these 3 sites and validated that 2 of them do, in fact, also produce viable drug-resistant viruses and are probably responsible for Tamiflu resistance today. So I'm bragging about this because this is probably the only time in my career that I will make a genuine computational prediction of any relevance to public health that is validated by someone who cares to do an experiment. So there are some practical benefits to understanding the structure of epistasis, I think. What I want to turn to now, and this is sort of a small change of pace, is I want to again study epistasis in protein evolution, but now in the context of a protein under purifying selection. The proteins we've looked at in this viral setting are continually adapting, but what about proteins that are simply being asked to do the same thing equally as well as have always done it over time? That's to say, proteins under purifying selection to maintain their structure, to maintain their function, and not to change at all. Is there any role for epistasis in this sort of very boring form of evolution? So to ask that question, we can't use empirical data like viral data, because we would need billions of years worth of data to have enough mutations to understand empirically what is the role of epistasis in purifying selection. So we took a simulation approach, choosing a particular protein, this ARG-T bacterial enzyme, that is small enough and whose crystal structure is known that we can, with great reliability, predict the energetic effects, the stability effects of a whole range of mutants in the wild-type form of the sequence. And so we have a fairly faithful in silico method based on a force field method to understand the stability effects of mutants in this very simple small protein. We can then imagine evolving this protein in a computer simulation, the core of which is this fairly complicated black box force field method of predicting the stability effects of mutants. We assign the wild type stability of the protein the highest fitness, and any versions of the protein that have lower stability or higher free folding energy are assigned lower fitness. And so this protein is simply under selection to maintain its stability, not to lose its stability, which is, in other words, this protein is under selection to remain the same. We then do a very simple sort of simulation version of evolution of this protein, introducing a whole range of mutations at any one point in time, assessing their effects on stability and their effects on fitness, and choosing one of those mutations to fix according to its relative fixation probability in a standard Moore-in-process model of evolution. And what we get out of this complicated simulation is just a series of substitutions in this protein, starting from the wild-type form. We get one substitution and a second substitution and so on, and all of these substitutions do nothing. All of these substitutions more or less don't change the stability of the protein very much, and they don't change the function of the protein. They've been selected by purifying selection to be the most innocuous substitutions for the most part. So it's in some sense a very boring computer simulation. And just to give you a feeling for what's happening in these simulations, if you look at a bunch of random mutations introduced into this wild-type protein, and you look at their selection coefficients, they're all negative because the wild-type has the highest fitness, and a huge proportion of them are lethal. So random mutations in the protein are typically lethal. They destabilize the protein. Nonetheless, the mutations that are actually selected by this evolutionary process are nearly neutral. They all have selection coefficients near 0, so the protein is just slightly changing its stability over time. Not much is happening. The fixed mutations, the actual substitutions that occur during this simulated evolutionary process, don't do much to the protein's stability or to the fitness of the protein. But we have this long chain of substitutions in a protein under purifying selection. And finally, we can ask the question I wanted to ask: what is the role of epistasis, even in this very sort of simple vanilla form of selection? Purifying selection is probably the most predominant form of selection, but it's typically neglected because it's so boring. Nonetheless, there might still be a role for epistasis. So for example, in this long sequence of different versions of the protein, imagine some focal substitution that occurred at some point in the evolutionary process, and we might ask, is there any way in which earlier substitutions modulated the presence of this focal substitution? Is there any sense in which earlier substitutions, this substitution was contingent upon earlier substitutions? And conversely, we can ask, once a substitution is actually made in a protein, are there any interactions that modulate what subsequent substitutions are allowed? So there might be a role for epistasis even in this very simple neutral walk. To measure that, we simply define an epistatic coefficient between a focal substitution I and some other set of substitutions j less than I or j even greater than I. And this definition of epistasis between the substitution number I and substitution number j is simply the standard definition of epistasis generalized to this long chain. And all it says is that we compare the fitness effect of the ith mutation at the time that it actually occurred to the fitness effect that that mutation would have had it occurred at some earlier time or had it occurred at some later time. So that's what these differences in log fitness effects mean. That we're just asking, how different is the fitness effect at the time of its occurrence in evolution versus had it been artificially introduced earlier or later in time? And the answer we find is that there's a substantial amount of both contingency and entrenchment in evolution under purifying selection. And this is sort of a very robust result with respect to how we fold the protein and what protein we choose. So let me just explain what I mean by this contingency and entrenchment. I'm looking at a protein that we've evolved for 30 substitutions, and we focus just by example at all of the substitutions that occurred at the 15th Markov step in this process across an ensemble of simulations. Now, at the time of their actual occurrence, when those mutations occurred at step 15, the fitness effects of those mutations were mostly neutral. They're near zero, which is something I've already told you. This is just a very boring process. And nonetheless, and here what I'm showing here in this contingency distribution is that had we introduced the very same mutations that actually occurred at the 15th step, had we introduced them in earlier genetic backgrounds, a large proportion of them would be effectively lethal. So there's a lot of, so what that means, what that implies is that although the mutations were neutral when they arrived at the 15th step, they would have been disallowed were it not for the presence of previous mutations often. And that's the sense in which protein evolution, even the simple protein evolution, is contingent on history. The earlier steps in its evolution sort of are the things that make possible the mutations that occur later. Conversely, we can ask, and this is actually an independent mathematical question, of the mutations that occur at step 15, when they occur, they're nearly neutral, of course. They don't really change the protein, which means that you could immediately revert them without any real effect on the protein. What would be the effect of reverting the mutations that occur at step 15 at later steps? And indeed, we find again that a large proportion of the substitutions that occurred at the 15th step are essentially lethal if we were trying to revert them later on in time. So this also has a strong sort of philosophical implication for evolution. It means that evolution is in some sense irreversible or rendered increasingly irreversible with time. An innocuous substitution occurs in the protein, we wait a little while, a few other substitutions occur, and then this innocuous thing we can no longer revert. It's now deadly to revert. So evolution is somehow channeled and sort of very rigid. Even this simple form of evolution is channeled and very rigid because of this sort of rich structure of epistasis that occurs within a protein. So I want to pause just to sort of think about why in the world these results are happening. That's to say, why is there epistasis in proteins under purifying selection at all? For those of you who know really even anything about protein folding, you'll know that the energetics of protein folding are well known and sort of a stereotypical example of an additive trait, a trait that doesn't have any epistasis at all. And in fact, that's true even in our own simulations. If you look at the energetic effects of pairs of mutations, the energetic effect of most pairs of random mutations are very well predicted by simply the sum of the energetic effects of the individual mutations. In other words, even in our simulations, this protein, this phenotype, protein stability, is essentially an additive phenotype that has very little epistasis involved. Nonetheless, we've observed tons of epistasis in evolution, and that's shown here. This is for random mutations. If you actually look at the mutations that are selected by purifying selection, and you introduce them in various combinations, they are not well predicted by their additive effects. There's indeed a substantial amount of epistasis between them. And so what I'm trying to say here, and this is a fundamental result in the sense that it has nothing to do with proteins per se, is that purifying selection enriches for epistasis. And this will be true for any fitness landscape and any phenotype that has even slight non-additivities, that if you select for a given trait value, you will enrich for epistatic dependencies between the mutations that accrue. And we can understand this mathematically, but I can also try to explain it to you intuitively. I almost want to just sort of step away from the podium. This is such a cool thing, and it's so easy to understand when you look at it in the right way. So why in the world does selection enrich for epistasis? And the answer has to do with regression to the mean, in a sense. Think about what types of substitutions would evolution permit? Well, we're permitting those substitutions that have no effect on the protein, more or less. So, well, what types of substitutions are like that? Well, first of all, if a substitution has no effect irregardless of the genetic background, irregardless of what other sites are doing, then that kind of substitution will certainly be accepted. But above and beyond that, if any substitution has a small effect and a yet smaller ameliorated effect because of the genetic background it happens to arise in, then that substitution will be preferentially chosen by purifying selection, right? Because we want to choose the substitutions that have no effect. And so again, purifying selection will enrich for substitutions whose effects are ameliorated by the particular genetic background in which they happen to arise, which means that the very same substitutions in different genetic backgrounds will have different effects, which is tantamount to saying that purifying selection will enrich for epistasis. And this is true for any phenotype. And it means that evolution has a very different view of the fitness landscape than the random mutations that are in the fitness landscape. Evolution really warps and enriches for these sort of strong interactions, even very simple forms of evolution. Okay, I want to switch gears now and talk for a little bit about actual organisms, or at least microorganisms, that are evolving. Some of you guys may know that there's a large and burgeoning field of experimental evolution where people observe evolution of organisms in action, mostly microorganisms in laboratory conditions. This field was really pioneered by Rich Lenski, shown here on the left, who 25 years ago or so now initiated 12 replicate populations, initially all the exact same genotype of E. coli, and has simply watched these populations evolve for the last 25 years. That's all he's done. But in many ways, I think that the impact of his work is tremendous. To me, he's sort of the modern microbial incarnation of the soul of Charles Darwin. What we've learned from these simple experiments are the beginnings of a real quantitative theory of adaptation. So for example, and before he did this experiment, I would have had no idea what to predict here. Over the first 10,000 generations of these E. coli, these bugs gained about 40% fitness, so that these cells are dividing about 40% faster today than they were when they started. So that's just to give you a feeling for timescale, how long it takes E. coli to adapt to, say, a laboratory condition, something I would have had no guess for whatsoever unless Rich had done this experiment. And this kind of thing is the kind of thing that Lenski and a bunch of, at this point, a huge cadre of experimental evolutionary biologists report. They do a replicate experiment across many populations, and they'll look at the pattern of fitness gains on average, averaging across the populations. And that's an object that a theoretician can certainly study. We call it the mean fitness trajectory. It is the expected fitness of a population at some time in the future, given that the population starts at a certain genotype on a fitness landscape at time 0. Of course, each one of many replicate populations will undergo some sort of stochastic process because of the forces of genetic drift, but the expected fitness is a deterministic object which has some value at some time in the future, and this is something that we can study. And so we've developed ways to parameterize large families of fitness landscape that encode different forms and different strengths of epistasis, and for each of these types of fitness landscapes, we can solve analytically, in fact, for the mean fitness trajectory, for the expected fitness as a function of time. And then finally, we can go back to the Lenski data and fit these trajectories to the data and find the best fit fitness landscape that explains the pattern of fitness gains that has occurred in these experiments. And in this example, the best fit fitness landscape among this big family is a fitness landscape which has a beta, which I haven't defined for you, which is less than -1, which means that there's a form of antagonism or diminishing returns epistasis in the E. coli landscape. So our inference here, looking only at the gross patterns of fitness gains in the Lenski experiments, was that the beneficial mutations in E. coli have a tendency to sort of disrupt each other's effects. They're antagonistic. They are less beneficial in combination than you would expect given their individual benefits introduced in the ancestral strain. So that was the inference we drew just from the pattern of adaptation. Fortunately, we convinced experimentalists to go and test this inference. So 2 experimental groups, Tim Cooper's group and Chris Marks's group, went and actually sequenced the ancestral strain, and they sequenced a derived strain much later. They looked at the mutations that had differed between the ancestor and the derived, and then they went back and they introduced these mutations in different combinations, and they directly verified that, in fact, in like 85% of the cases, these combinations of mutations are less beneficial, substantially less beneficial in combination than you'd expect under independence. And so there's sort of an antagonistic epistasis that is somehow slowing down the evolution of these prokaryotes. So at this point in time, we were feeling really good about ourselves. We had made a prediction from the sort of gross aspects of evolution in this experiment, and then experimentalists had gone and taken the time to actually test it and, for the most part, actually validated the prediction. What I want to spend the last part of my talk talking about is a series of complications that make us feel less good about ourselves, but are actually quite interesting. Things that complicate this issue of inferring the fitness landscape from evolution experiments substantially. So I will skip this one and start right in on the so-called complications that arise from problems of identifiability. So we asked ourselves more recently, under what conditions, what kind of observations can an experimentalist make that allow them to genuinely infer the presence of epistasis, which is certainly a prerequisite to inferring the form and shape of epistasis in a fitness landscape. So what I mean to say is, we ourselves in the past, and more recently Rich Lenski himself, has looked at, in this case, 50,000 generations of the average, shown in white dots here, patterns of fitness gains. And we have inferred that there's antagonistic or diminishing returns epistasis in the E. coli genome. So in particular, Rich Lenski most recently fit a power law model to this pattern of fitness gains and described that this power law is precisely what you would expect if there's antagonistic epistasis, that mutations disrupt each other's beneficial effects. So this is all well and good. This is, in fact, exactly what we did as well. The problem we've come to realize is that we can equally well fit this precise pattern of fitness gains with a completely non-epistatic fitness landscape where there's simply independent effects at different sites. So we can get a perfectly good fit that's statistically indistinguishable with a very simple model that doesn't have any epistasis at all. And what's happening here, the problem that's happening here, is an illustration of a much more general feature. In fact, we've proven this horrible theorem that says, under certain technical conditions that I'm not going to go over but aren't really all that restrictive, that any mean fitness trajectory, any pattern of fitness gains that can be produced by an epistatic fitness landscape that has dependencies can also be produced by a non-epistatic fitness landscape. So whatever pattern of fitness gains you observe, you can spoof it by constructing, this is a constructive proof, a non-epistatic landscape that produces the exact same pattern. So let me just give you one more example of that. This is just a toy example, but it's a fun one. Imagine a fitness landscape that has only 2 sites, A and B. All the populations start at this initial genotype. They'd like to go to this highest peak, but to do so they have to cross a valley. So the typical fitness gains in such a population, the expected fitness over time, I hope you can see it here, is actually a complicated non-monotonic curve. It starts here at log fitness 0. It goes down because populations have to cross this valley, and eventually it goes back up as most populations have crossed this valley and reached a high fitness. So this is a fitness landscape that exhibits so-called sign epistasis, and it has a very complicated fitness trajectory. Unfortunately, we can spoof this fitness trajectory by a simple non-epistatic fitness landscape that has 3 sites. Each of them have completely independent effects on fitness. And this landscape provably produce the exact same mean fitness trajectory. And so these are, again, just 2 examples of this general theorem which robs us of the ability to infer epistasis from observing the mean fitness gains in a population. This is a sort of non-identifiability theorem. It tells us that it is simply impossible to infer the presence, let alone the form of epistasis, by observation of the pattern of fitness gains in a bunch of replicate populations, despite the fact that we have made those inferences in the past, and others as well, and many others as well. This theorem tells us that in principle, if you consider all fitness landscapes, you cannot make such an inference. And it gets worse, actually. You might think that, well, okay, experimentalists, they don't look at fitness alone, they can also look at mutations. They can count how many mutations have accrued over time, what we would call the mean substitution trajectory. But we've also proven this result, that the mean fitness and the mean substitution trajectories can be varied independently of one another. And so what that means is that their joint observation, observing both the mean fitness gains and the mean number of substitutions that have fixed, mutations that have fixed, doesn't actually increase your inferential power at all. And so it's not as if you learn more by observing both these things, which is what the experimentalists would naturally like to use to make some inferences. And so we have these unfortunate disastrous results that say that it's, in principle even, it's impossible to draw inferences from the mean pattern of adaptation. Okay, I don't ever want to end on this negative note, and so I will tell you a few positive results with some glimmer of hope for the future of microbial evolution. And the most obvious thing to do is to look at the variance. So, of course, these experimentalists, Lenski started 12 replicate lines. Nowadays, there's a whole number of experiments with 10,000 replicate lines that are evolving in a lab. And the obvious thing to do, and in fact, if you look at Rich Lenski's initial 1994 paper where he initiated this whole program of looking at evolution in a lab, he said that I want to start 12 replicate lines all from the exact same starting genotype because I'm specifically interested in what differences occur between the different populations. Because presumably, if the fitness landscape is rough, then some of these populations will go down one avenue and other populations will go down a different avenue. And so this is the resulting ensemble variance trajectory that will typically occur. Over short times, first of all, at time 0, there's no variance in fitness across the populations because they're all the exact same genotype. But over short time, there will be a rapid increase in variance as some populations go in some directions, other populations go in other directions. Over very long times, however, the populations will tend to approach the global maximal fitness in the landscape, and so the variance should decrease. So just roughly speaking, this is what the variance trajectory should look like. And Lenski himself said that ideally looking at the variance will tell us something about what forces are driving evolution in these E. coli. And now, fortunately, he's provably correct. It turns out that, and we can prove that non-epistatic landscapes will always produce variance trajectories that are concave at short times, whereas there are some epistatic fitness landscapes that can produce convex variance trajectories. And so what this implies, of course, is that it is possible to infer the presence and, in fact, the form of epistasis by looking at the variance in the evolutionary course of a bunch of replicate populations. And let me just give you a feeling for what causes this. If there's a kind of epistasis whereby one neutral mutation is a required prerequisite to adaptation, so there's a fitness landscape where you need a neutral mutation that doesn't change fitness, and thereafter you can get a beneficial mutation, then that will produce this sort of convex up accelerating variance across populations. So in particular, this means that the exact kind of mutation that Jesse Bloom and David Baltimore found in influenza virus, a neutral mutation that is a prerequisite for a resistance site to be useful, would produce this type of convexity in variances across replicate populations. So exactly the form of epistasis that's been observed to be important to the adaptation of microbes is fortunately the kind of epistasis that will allow us to actually to infer the form of the fitness landscape. So that's good news. There are a whole range of other things that can allow you to infer epistasis from patterns of evolution, which I'm not going to go into in much detail. Let me just summarize them here, just to give you a feeling for sort of the directions of a research program here that I'm very interested in, which is to work out systematically all of these questions of identifiability, to ask what types of observables can experimentalists look at, which will actually provide them, even in principle, any inferential power to learn what it is about the fitness landscape that guides evolution. So we've seen, for example, that the mean fitness over time does not provide any power, that's why I have an X here, to infer even the presence of epistasis, whereas the variance trajectory over time does. There's a whole range of other things which are somewhat surprising, like the equilibrium time, the time to stationarity, also cannot provide any information about whether or not there's dependence or independence of sites, whether or not there's epistasis. A whole range of things lack information. But fortunately, a few things do contain information. The most informative, actually, is the so-called time evolution of the DFE, the distribution of fitness effects. For example, if you measure in the ancestral strain, what are the fitness effects of a bunch of random mutations? And then you let that strain evolve for a while, and you measure again, what's the effects of a bunch of random mutations? The change in the DFE is provably sensitive and in fact highly powerful for inferring the form of epistasis that's in that landscape. And so that's a very reasonable thing that's actually fairly cheap for the experimentalists to measure that will allow them to understand what's actually guiding evolution in their experiments. So I want to close there really by thanking once again the Society for awarding me this wonderful Akira Okubo Prize. I have to say I'm very honored by the confidence of the prize committee, but especially honored to be associated with Okubo's name. More important than that, though, I really want to thank the Society, probably from all of us, for something that they do tirelessly and usually without recognition, which is promoting the importance and excitement of mathematical biology across the globe. Their work benefits all of us, collectively and individually, and so I'm just thankful for the existence of SMB, and especially to the dedication of its officers. So thank you to the Society. Also, I can't end without thanking a few other people. I also want to thank my two graduate supervisors, Simon Levin and Martin Novak. If any of you guys know them, then you already know how incredibly lucky I was to have these two as my graduate mentors. They're both remarkable scientists in their own right, but they're also very different scientists. So from Simon, for example, I learned a large number of good jokes as a PhD student. And from Martin, I learned an equal number of bad jokes, amazingly. But in each of them, I got a model for what it is to live life driven by curiosity and by the joy of sharing your curiosity with others. And for that, I am always grateful to them. And lastly, and really certainly the most important thing I want to do is thank all of the students and postdocs that I've had in my research group over the past 8 years. So these students sort of regularly expose me to all sorts of fascinating ideas, scientific and otherwise. And indeed, the very ideas I presented today are due to these students. And so my students do no less than enrich my intellectual life and the lives of each other every day. And for that, I will be eternally in their debt and always their strongest supporter. So finally, thank you guys all for the invitation to speak and for your attention. I'm happy to take any questions you might have.
About the last part. So you started with showing there are individual trajectories. So wouldn't and the mean and the variance are just two moments, essential of a fitness. But then you have actual trajectories. Is that informative in any way?
Oh, that's a
like
great question.
beyond just 2 moments of mean and variance. Kind of.
Well,
can we tell epistasis from individual trajectories?
Good
Their
question. So
divergences.
the question was, can you, yeah, so I guess it was a microphone. The answer is yes. Even from a single population observed for a sufficiently long period of time, you can, in fact, that a single population observed for sufficiently long, once it reaches equilibrium actually, does have information about the presence of epistasis. In particular, reversion rates. So if a mutation occurs at a particular site, if there's no epistasis, the rate of reversion is just, the hazard function of reversion is constant in time. It doesn't depend upon any other mutations. But if there's any epistasis for that site, then the rate of reversion will be strictly decreasing in time. So even a single population observed for sufficiently long would contain information. But I should say that those are impractical amounts of time. So experimentalists prefer to take 10,000 populations and evolve them for a year. And they need to learn what moments or what other statistics from their data should they look at to make any inference. So a single population in principle is helpful, but I think in practice isn't really going to be useful. Well, under certain reversibility conditions, there's always an equilibrium, but yeah.
Hello. Thank you very much for your talk. I really liked your talk, and my question is, will you expect to see diminishing returns epistasis during the clonal evolution of eukaryote populations?
Can you just repeat, in what kind of populations?
Eukaryotes?
Eukaryotes. Oh, yes. So actually, there's been an experiment already in budding yeast, Saccharomyces yeast, where they were haploid budding yeasts, but they're eukaryotic organisms, and they also exhibited this pattern of diminishing returns epistasis. In the sense that that the experimentalists actually introduced mutations and combinations and directly measured that there's diminishing returns epistasis. So yes, it has occurred in other microorganisms as well.
For instance, I mean, in cancer progression, right? The type of epistasis, do you think that the interaction among beneficial mutation would be diminishing returns?
No. In fact, I skipped over the slide, but the predominant form of epistasis that you expect to find depends upon whether or not the population is saturating in fitness. So very far from the fitness peaks, and cancer populations are still rapidly acquiring beneficial mutations, there will be a prevalence of synergism. It's only it's only as you approach fitness peaks that the form of epistasis changes. So I do not actually think that in a somatic hypermutating or cancerous population that there will necessarily be antagonism amongst the mutations that are accruing. So, but I don't actually know. I mean, that experiment would be wonderful to do, and I don't think it's actually been done, introducing or co-segregating mutations that occurred at different time points during tumor progression.
Josh, thank you for a wonderful talk. I think Akira would have been really thrilled to have heard it. Unfortunately, he couldn't. But a predecessor of yours at Pennsylvania, Stu Kaufman, who was also a president of this society before Simon, has built recently what I guess you could call a kind of theory that he calls the adjacent possible. And I don't know if you're familiar with this at all. But in typical Stu form, it's a general idea that we cannot readily predict because of the historical nature of evolution what will occur. Just as 50 years ago we could not have readily predicted Google, or at least if we could have made a lot of money. And we've published a couple of papers that show, in some sense, that this is a computationally irreducible problem in the Steve Wolfram notion of computational irreducibility. So my question is, you have given me some hope that indeed there is some capability of looking at epistatic interactions and predicting, to some extent, things that Stu would call adjacent possible. So I'm going to encourage you, if you have not looked at this stuff, to do so, but I'm also interested in whether or not you have any thoughts on that.
Well, I have. I've thought a lot about the prospects for predicting molecular evolutionary courses, especially in viral settings. So, and I have to say I have mixed feelings on that. In not the technical sense that you're describing, computational irreducibility, there is a sense in which some of these results say that evolution is quite hard to predict. The degree of contingency that we observed in purifying selection, what that means is that you cannot simply assay the effects of mutations in an ancestral strain and say, oh, well, this mutation is useful, so it's eventually going to occur. Because at the time of its occurrence, other mutations will have occurred in between, and they will modulate the selective consequences of a focal mutation. And that contingency is very strong. So that leads to a certain form of unpredictability. At the same time, in the context of flu viral proteins that are adapting, we have demonstrably understood retrospectively dependencies that allowed us to make genuine predictions for the future that were validated. So in some sense, on short time scales, when hopefully the fitness landscape isn't changing too much, we can make some predictions. But again, I have mixed mixed feelings about the prospects of prediction.
Other questions?
I'm a little kind of surprised and amazed that in your simulations with the protein sequence, essential evolution didn't really do much, so the average fitness didn't increase. I wonder if, and it's like kind of contradictory in some way, counterintuitive, in a way that evolution should increase fitness. I wonder if it's that because you started with a good sequence.
Oh,
if you were to start it with a poor sequence, which we would think that most evolution starts with a bad one, then you would see a lot of improvements over time.
Oh,
and then it will saturate, of course, depending on the way.
Certainly. We specifically chose the wild-type sequence, which has presumably been, over billions of years, selected to be stable, because we wanted to study, in that study, the effects of purifying selection alone. We didn't want to focus on the adaptation process. But you're right, of course, if we started it far from the wild type, and actually we did simulations like that too, it increases in fitness.
Okay, well, thanks, Josh. That was a spectacular talk.
Thank you.